In the second half of this lecture, we'll cover the effects of using hops. At the beginning of the lecture, I mentioned that hops have bacteriostatic activity. This slide simply shows how increasing concentrations of trans-isohumulone inhibits microbial growth, in this case the growth of lactic acid bacteria. Recall as well that this antimicrobial activity, while perhaps less important in the modern brewery, may have been important in establishing hops as a basic beer ingredient in the breweries of the past, where spoilage is a big problem. Simply notice that at high concentration, bacterial growth is very low and increases with decreasing concentrations of the trans-isohumulone. Though there are certainly flavor and other advantages to using hops in beer, let's turn to some of their associated problems. Riboflavin readily forms an electron scavenging triplet state upon irradiation with visible light. This species takes an electron from that alpha acid's same delocalized electron density that was responsible for the formation of the isoforms in the first place. Riboflavin's electron scavenging leaves the hop acid a radical, and intramolecular rearrangement leads to cleavage of the acyl chain and formation of a ketyl-acyl radical pair. Subsequent loss of carbon monoxide from the acyl radical and recombination of the resulting fragment with a thiol radical produces 3-methyl-2-butene-1-thiol, also known as skunky thiol, as well as dihydrohumulinic acid. Note the extremely low perception threshold in the parts per trillion of this molecule. Since riboflavin is activated by light in the first place, storage of beer in brown bottles is a highly effective means of preventing this particular off-flavor's development. Another problem with hops and flavor stability is the formation of tricycloderivatives. First, let's notice again the different chemistry of the trans on the left side and cis isoacids. Their tricyclo products lead to weaker bitterness, but also to an alteration of the character of the bitterness, with most tasters considering the flavor to be more harsh. It's worth pointing out here that though we often dismiss taste as being simple, with only 5 possibilities, sweet, sour, salty, umami, and bitter, in fact, humans have 25 different taste receptors just for bitterness, and so our perception of bitterness is not a singular thing, but much more subtle, perhaps accounting for the big difference in perception of the isoacids and their tricyclodegradation products. Anyway, it's the trans form of isoacids that are most susceptible to further cyclization and which support the formation of a few different compounds. The details of their structures are not so important to us, but showing them to you with this color code makes it easier to understand the figure on the following page. Tricyclohumol in red, tricyclohumine in yellow, isotricyclohumine in purple, with the trans and cis forms of isohumulone in yellow and gray. This chart is from a wonderful and comprehensive analysis of hop degradation and shows the concentration of isoacids and their tricyclodegradation products as a function of storage time. Remember that in yellow and gray we see trans and cis isohumulone, while the cis form does decrease with time by some 30%. The trans form is 90% consumed over the same period. At the same time, tricyclohumol increases dramatically. Why is the trans form less stable? It's always about sterics and electrostatics. In this case, after the acyl carbonyl accepts a proton and forms a carbocation, in the trans isomer, the side chain at carbon 5 has a double bond which is positioned for nucleophilic attack on the hydroxyl carbocation. The cis arrangement doesn't present side chains appropriately, so the trans isomers are the more favorable. Okay, I got a little wild with this slide, but many brewers wish to take advantage of the gentler temperature conditions post-boil to extract volatile compounds like the beta acids that might otherwise degrade or volatilize during the boil. So you get it, mo beta. Anyway, this is called dry hopping. There are some downsides that come with dry hopping, however, that can lead to flavor or other problems. First, yes, there are stronger aroma contributions, especially of terpenes like linalool and geraniol, and the alpha acids shouldn't isomerize as much at lower temperature. However, dry hop beers often have an enhanced bitterness. There is, in fact, some amount of isomerization, but dry hopping tends to extract still another class of hop acid degradation products called humulinones, which are nearly 70% as bitter as isoacids themselves. These are not produced in boiling, however, and are actually degraded during the boil. Another challenge is hop creep, or the increase in ABV upon dry hopping. The figure on the right illustrates this. The ABV creep occurs for a number of possible reasons, including the fact that unpasteurized hops will undoubtedly bring with them wild yeasts that continue fermentation. But what sugars will they ferment? Well, hops are plant material and have about 2% monosaccharide, so they bring fermentables to the table. Not only that, hops are themselves diastatic, with many of the same amylases present in malted barley, so hops can degrade their own starches into still more fermentable sugars. All this hop instability I've described means some brewers look to addition of more sophisticated hop derivatives as a solution. These di-, tetra-, and hexahydroisohumulones are chemically modified and might be added to the beer after fermentation. Reduction of unsaturated carbons in the isoacid's carbon-5 side chain improves their stability both to radical cleavage and skunky flavor development, and to tricycloderivative formation. Recall that no carbonyl at the 4 position means no carbocation formation and no subsequent attack by carbon-5 side chain. Likewise, reduction of the unsaturated carbons in the terpenoid side chains correlates with a more foam-positive trait, that is to say, a more stable foam head. Here's a bitter pill for you. How does this chemical modification of something that's usually consumed as a raw ingredient affect how you feel about the product? Would hyperprocessing of this type to serve the needs of a large-scale brewing to have a more stable and consistent product be better or worse than a more natural product that is prone to off-flavor development? Let's turn for a moment to the traits of individual hop varieties, or cultivars. This image is taken from the Homebrewer's Formulary poster that's floating around the internet. 30 or so common varieties are shown here with their typical range of alpha acids listed. I guess what's most interesting here are the outliers, with Crystal lying at the low end of the alpha acid concentration and thus among the aroma hops on the left, while Chinook, Columbus, Eroica, Galena, and Nugget all reside in the bittering hops category on the right, and the extremes might be 18% alpha acid by weight. This figure is from Brewing Classic Styles by Zainasheff and Palmer, and is reminiscent of the common beer flavor wheel I showed you in the yeast lecture. I guess I'd like you to see the spectrum of aromas here: floral, fruity, citrus, herbal, grassy, evergreen, and spicy. For context, let's highlight the so-called noble hops of European lager fame, which on this chart straddle a range of traits, though they're often described as spicy. Also, referring back to where we started in this lecture, Saaz, originally a Czech hop, are now grown in New Zealand as well, and the new hops are considered to have characteristics distinct from the old, tending more to citrus than their European progenitors. You might use this as a general guide to extremes if the goal is to isolate a particular aroma in the beer. Simcoe, for example, is recognized as particularly piney. Finally, let's highlight Cascade as it comes up on the very next slide. The hop aroma diagram on the previous page makes clear that the taster perceives very different aromas in different hop cultivars. Those distinctions can be harder to quantify analytically. The molecules are easy to detect, but there's so much interaction between them it can be difficult to establish what might be dominant to an individual. This study looked at aroma molecules across a range of hop cultivars. The easiest trend to identify was the presence of a set of terpenes across all hops. Myrcene, linalool, and geraniol, all diterpenes, were found at high concentration in all hops tested. Cascade hops were found to be high in nonanol, so in some cases, individual hops do have a distinctive flavor molecule that corresponds with their flavor descriptors. Finally, I like the symmetry of closing the lecture by coming around to not a phylogeny like I used to relate the hop genus to other flowering plants, but a model generated in a very similar fashion. This is a dendrogram of hop relationships built not from genomic sequences, but from percentage composition of volatile oils. The compositions cluster these hop cultivars more or less as one would expect. First, there's variation among individuals. Note that the Saz samples diverge from one another, sometimes dramatically. Second, there's order. Yes, the noble hops fall into a single group, while Cascade, Willamette, and the rest fall into another. But recall that this organization is based not on preconceptions or prior knowledge, but on volatiles composition. Here too, though, the clustering does make sense, with noble hops, known for their spicy aroma, being high in spicy sesquiterpenoids such as humulene, while hops known for their citrus odor being high in monoterpenoids such as geraniol.